Hi all, I'm Gayathri Sambamoorthy from the Indian Institute of Technology, Madras. I'll be talking about my research on understanding the evolutionary dynamics in microbial communities. So to start off with, I'll give a brief introduction. Microbes live in a wide range of habitats and often they exist in microbial communities by interacting with one another by the exchange of metabolites. These metabolites are more often called the cross-fed metabolites. And the type of interaction may vary. They can be beneficial, harmful, or neutral. And based on the way that they interact with one another, they can be classified into different types. Amensalism, where one organism has a negative effect on the other, and commensals are the one where one organism has a positive effect on the other. Mutualism, where both organisms benefit from each other. Parasitism is where one organism has a positive effect and the other has a negative effect. And competition is where both organisms compete for one another. And neutralism is where there is no gain or harm with respect to the microbes in the communities. So the motivation for our study is that there have been several studies to understand the natural communities and also how synthetic communities can be employed to co-produce biomolecules. And co-evolution has been shown to change the dynamics of the microbial communities. So what we are looking at is how does the dynamics of the microbial community change upon co-evolution. And we wanted to answer a few key questions. Do interaction patterns remain the same upon co-evolution? And does co-evolution confer fitness benefits? And how does the evolutionary pressure shape microbial interactions? So we answer these questions using metabolic networks as model systems. We picked microorganisms, the genome scale metabolic networks from human gut microbiome. And of these organisms, we form two-member communities that exhibit different interaction patterns, and also formed a universe set of reactions from all the reactions in the Agora database. We employed three different constraints for co-evolution. The first constraint is where the combined growth of both the organism is greater than 50%. In the second, it's greater than 10% of the wild type or the starting community. And the third is where we don't have a constraint on the combined growth, but in all three cases, we have a meager individual growth of organisms that needs to be sustained. And the methodology for co-evolution is we have a community of microorganisms and we pick one reaction at random from these community and swap it with one reaction from the universe set of reactions. Once done, we check for the growth constraints and accept if the growth constraints are met and reject if the growth constraints are not met. We repeat the process 1,000 iterations and we identify one co-evolved communities. And by this process, we evolve 1,000 co-evolved communities for one community. We basically employ community flux balance analysis to identify the growth phenotypes. So in total, we have 90,000 co-evolved communities for 6 interaction types, 5 different communities for each interaction type, and they've been evolved in 3 different constraints. So totally, we have 90,000 co-evolved communities. So what we went ahead to do is to understand how the interaction patterns of the co-evolved communities vary. And using the constraint 1, what we observed is that the major fraction of the co-evolved communities, starting from amensal, commensal, competitive, and neutralistic pairs, was parasitic, whereas mutualistic and parasitic communities yielded more of mutualistic communities. With respect to the constraint 2 and 3, we had similar observations where all the communities, irrespective of what interaction type they started off with, they ended up in a larger fraction of parasitic communities. Next, we went ahead to see if there are growth capabilities or growth enhancements upon co-evolution. And this is basically a plot of the distribution of the increase in the combined growth of the organisms in the community after they've evolved with the initial growth in the community. So These are the wild type communities. So how have the growth increased if they've started off as an amensal and evolved as a amensal, commensal, and so on. So in all cases, we see that the mean of the growth is higher for competitive communities, irrespective of what community you started off with. Another observation that we saw is with respect to the metabolites that have been exchanged. So we see that the number of metabolites that have been exchanged in mutualistic evolved communities are much higher than the other interaction types, irrespective of what interaction type we started off with. So to summarize, coevolution alters interaction patterns. So the interactions are not stable, and you start off with some interaction, you end up in different interaction type. And many co-evolved communities showed a reduction in growth, and some of them showed an increase in growth as well. And there are competitive communities which show an increase in the metabolite exchanges. And the mutualistic pairs show higher number of metabolites being exchanged when compared to the other communities. So overall, our study throws light into the important aspects of microbial coevolution and the stability of microbial interactions. Thank you.